So, good morning all of you. Welcome to the video lecture on Cilia and Flagella. You have heard about Cilia and Flagella, isn't it? Especially during the when you studied about the bacterial structure, isn't it? Cilia and Flagella. So, let us discuss about the structure of Cilia and Flagella. First, let us know what Cilia and Flagella are. As far as cilia and flagella are concerned, these are hair-like outgrowths which develop from the cell membrane, hair-like outgrowths of cell membrane. Now, cilia and flagella, they differ from each other in terms of size. Well, cilia are smaller in size and they are more in number, whereas flagella are more elongated but fewer in number. Okay. Now, the cilia are present on entire surface of the cell, whereas the flagella are generally found or present on the anterior side of the cell. Okay, generally, I am saying generally. Now, cilia beat in a synchronous manner, whereas flagella, they show independent movement. So, these are the basic difference between cilia and flagella. So, cilia are smaller and more in number, whereas flagella are more elongated and fewer in number. Cilia are present on entire surface of the cell, whereas the flagella are present at the anterior side. Cilia beat in a synchronous manner, whereas flagella show independent movement. Now, let us come to the structure of cilia and flagella. As far as cilia and flagella structure is concerned, there is presence of a central bundle of microtubules which are called axoneme or shaft. Okay, presence of central axoneme or shaft. Now, axoneme is made up of 9 peripheral doublet microtubules. Okay, 9 peripheral doublet microtubules with central singlet microtubule. Okay, I will show that in diagram. So, the presence of 9 peripheral doublet microtubule with central singlet microtubule. So, this type of arrangement is called as 9+2 arrangement. Got it? Now, let us come to this peripheral one. You know that peripheral ones are the doublet microtubules. Now, each peripheral microtubule is made up of 2 tubules, because we said that these are doublet. So, these are made up of 2 tubules A and B. Okay, we name it as A tubule and B tubule. The A tubule is made up of 13 protofilaments, whereas B tubule is made up of 10 protofilaments. You would be able to understand it while discussing about it in diagram. Okay, I will show the diagram. You will completely understand regarding the structure of axoneme. Okay, so axoneme is made up of central portion of a singlet microtubule and the peripheral of your doublet microtubules, then pair of singlet microtubules at the center and peripheral microtubules, 9 peripheral microtubules which are called as 9+2 arrangement. The peripheral doublet microtubules, central one is singlet and the peripheral ones are doublet. Doublet microtubules consists of A tubule and B tubule. The A tubule is made up of 13 protofilaments, whereas the B tubule is made up of 10 protofilaments. Now, these axoneme is held together by 3 sets of protein crosslinks. Okay, 3 sets of protein crosslinks fix or hold this axoneme together. What are those? The first one, let us consider about the first one. The central portion is occupied by 2 or pair of singlet microtubules. I have already discussed about it. Now, as far as this central singlet microtubules are concerned, they are joined by a bridge and are surrounded by a fibrous structure called inner sheath. Okay. So, singlet microtubule, they are joined by means of a bridge and surrounded by a fibrous structure called inner sheath. Now, the second set of linkers are made up of nexine proteins. Okay. Second set of linkers are made up of Nexene proteins that join the outer set of doublet microtubules. Okay. That join the outer set of doublet microtubules. That means doublet microtubules are joined with each other through Nexene proteins. Got it? Then the third linkers or third set of linkers are actually radial spokes that connect the central singlet, okay, the central singlet microtubule with each of the A tubule of outer doublet. Got it? So, 3 protein cross links are there. The central singlet microtubules are joined by means of a bridge and they remain surrounded by means of inner sheath first. Second one is the peripheral doublet microtubules are joined with each other by means of Nexene protein, and the third linker is radial spoke, rather are radial spokes, which connect the central singlets with each of the A tubule of outer doublet, each of the A tubule of outer doublet. Got it? So, now the doublet microtubules, which are the peripheral ones, they are made up of specialized proteins, protein polymers called tubulin. Okay, they are called tubulin. A tubule of doublet has inner and outer rows of arms that I will show in diagram in the last slide. So, each A tubule of a doublet has inner and outer rows of arms made up of dynein protein. Got it? Which are involved in ATPase activity. Okay. Now, let us come to the functions of cilia and flagella. You know it very well. Help in locomotion, then they create water and food currents, then help in the elimination of solid particles. These are very simple functions. Now, come back to the structure. As far as the diagram is concerned, you can see that there is presence of your peripheral, this one is the peripheral doublet microtubule and this one is the central pair of singlet microtubules. Got it? Now, as far as the peripheral ones are concerned, your each one is made up of each doublet microtubule is made up of one A tubule made up of 13 protofilaments and one B tubule made up of this one is A tubule made up of 10 protofilaments. Got it? Now, these 9 doublet peripheral microtubules are there, isn't it? I have already told you 9 are there at the periphery. Now, these doublet microtubules are joined with each other through Nexene proteins, as you can see here, Nexene protein joins it. Got it? Now, another thing we discussed that the peripheral doublet microtubules, out of which the A tubule is also linked to the central singlet microtubule by means of radial spokes. What are the radial spokes? This one is the radial spokes. So, it is connected with it. Got it? It is connected with it. As you can see in the second picture here, this is the radial spoke, this is the radial spoke, this one is the radial spoke. These radial spokes are joined with the singlet central microtubules join the A tubule to the singlet microtubule at the center through radial spokes. Okay. Now, let us come to the your central one. There is presence of central pair of singlet microtubules. This one is a microtubule, this one is a microtubule, and these are joined by means of a bridge. You can see in this particular picture, probably this one is the picture. Okay, let me tell you, this one is the bridge, this one is the bridge. Got it? And another thing that I have told you, that the central singlet microtubule that remains surrounded by an inner sheath. Here this is the inner sheath. Okay. So, this is what the simple structure of cilia and flagella. Now, I have discussed about another thing, that the air tubule remains associated with another dynine arms. Okay, I have already discussed that it is associated with dynine arm, and these are concerned with ATPase activity. Got it? So, this is what the structure of cilia and flagella, very simple structure. I hope you would be able to understand it. Got it? So, thank you very much.